Hello everyone, today we are going to learn in vitro hemocompatibility test. In today's lecture, we will be learning about what is biomaterial and biocompatibility. We will be also learning various hemocompatibility tests. Under the hemocompatibility test, we will be learning hemolysis assay more in detail, both theoretically as well as practically through demonstration. Let us see what is biomaterial. Biomaterial means any material that has been designed to interact with the biological system for medical purpose. It can be for therapeutic or it can be for diagnostic application. Biomaterial is used to replace a part of a function of the body in a safe, reliable, economic and physiologically acceptable manner. So, these are some of the example of biomaterial: dental implants, joint replacements, hip replacement. These are some of the examples for the biomaterials. Whenever we develop the biomaterials, we have to understand the biocompatibility before we implant these biomaterials into the human body. Let us see what is biocompatibility. Biocompatibility means material's ability to function properly with a suitable response from the body in a given application. The material should be compatible to the biological system. It should not induce any adverse effects. That is called biocompatibility. The biocompatibility of biomaterials can be assessed and tested by the following criteria. The first one is genotoxicity. Genotoxicity means the particular material should not induce any damage to the DNA. Next one is cytotoxicity. The particular material should not induce any toxicity to the cells. Third one is immunogenicity. The particular material should not induce any immune responses including allergic responses. The fourth one is hemocompatibility. The particular biomaterial should be compatible to our blood, that is called hemocompatibility. Let us see why to do hemocompatibility test. Hemocompatibility test is very important for biomedical devices and implants that interact with the blood because it focuses on unique risks such as thrombosis, hemolysis and immune reactions. So, that is why the hemocompatibility test is very important to understand the safety and efficacy of the blood contacting materials. The other biocompatibility test mainly focus on the general safety of the particular material, but the hemocompatibility mainly focus on the interaction between the materials and the circulatory system. So, that is why this hemocompatibility test is very important for implantable biomedical devices. That is the reason the hemocompatibility is one of the important factor in deciding the application of implantable biomaterials such as artificial blood vessels and orthopedic implants. Whenever we develop biomedical implants, we have to study the hemocompatibility, and if the hemocompatibility is not good, we have to improve the hemocompatibility by surface modification or redesigning the particular material before we implant into the body. According to International Organization for Standardization, the hemocompatibility tests are conducted in the following model. The first one is static. Under the static, we have blood coagulation assay, hemolysis assay and partial thromboblast in time. Under dynamic model, we have Chandler loop system and chronic shunting. In today's lecture, we are going to learn hemolysis assay more in detail. Before we learn about hemolysis assay more in detail, let us see what are the various hemocompatibility tests. Under the hemocompatibility test, there are various assays. Let us see one by one in detail. The first one is hemolysis assay. This assay is to evaluate the damage to red blood cells after exposure to the test material. The test material can be a biomaterial or it can be a nanomaterial or any kind of test substance. And next one is anticoagulants. This assay is to measure the concentration or activity of anticoagulant drugs in the blood. And third one is platelet adhesion and activation. This assay is to evaluate the impact of test materials on blood platelet adhesion and activation. By this assay, we can understand whether the platelet is attaching to the biomaterial and is there any change in the shape. So, if there is a change in the shape and if the platelet is attached to the particular material, that material is not suitable for biomedical implants or clinical application. And next one is blood coagulation time. So, this assay is to measure how long it takes for blood to clot in presence of test material. And the last one is blood protein adsorption assay. This assay is to quantify the amount of blood proteins adsorbed to a surface. The surface can be a biomedical implant or medical devices. Let us learn about hemolysis assay more in detail. When RBC, that is red blood cells comes in contact with the test material, that is your nanomaterial, this nanomaterial disrupt the membrane integrity of your RBC. Once it damages the RBC membrane, hemoglobin in the RBC will leak out of the cells. That is called hemolysis. The degree of hemoglobin release calculated in terms of percentage to understand the hemocompatibility of nanomaterials. And this is a simple and cost effective technique. That is why most of the lab they use this hemolysis assay for understanding the hemocompatibility of their biomaterial. Let us learn how to perform hemolysis assay. The first step in the hemolysis assay is procurement and processing of blood. We can collect the blood from human or goat. We have to collect the blood in a container which contains tripotassium EDTA, which is an anticoagulant. And very important point is we have to take the approval from the Human Ethics Committee or Animal Ethics Committee or Institute Biosafety Committee for handling certain samples. Before you handle some of the blood samples or any sample which is related to human or animal, we have to take approval from the particular committee. Once you collect the blood, the next step is you have to centrifuge the blood at 2000 rpm for 10 minutes, then isolate the RBC. Once you isolate the RBC, we have to add the colloidal nanomaterial of different concentration for understanding at what concentration the nanomaterial is inducing the hemolysis. Once you add the nanomaterials, incubate the RBC with nanomaterials for 2 to 3 hours. After incubation, you can do the centrifugation. Once you do the centrifugation, if the particular nanomaterial is inducing hemolysis, you will get this kind of result, and if the particular nanomaterial is not inducing any hemolysis, you will get this kind of intact pellet. Then you collect the supernatant and measure the absorbance using spectrophotometer. The absorbance can be measured at 541 nanometer using the plate reader. Then the percentage of hemolysis can be calculated by A test minus A negative divided by A positive minus A negative multiplied by 100%. A test means this is the absorbance of supernatant from the test samples, and A negative is absorbance of supernatant from the negative control tubes, and A positive is the absorbance of supernatant from the positive control tube. Here this negative control is we will be using only saline, and in the positive control we will be using water, or we can use some kind of detergent which can completely damage the RBC membrane and it can release all the hemoglobin. That is your positive control tube. And once you plot the hemolysis percentage, you get this kind of data. From this data we can understand whether the particular material is hemolytic or not. For example, if the hemolytic percentage is between 0 to 2%, it is non-hemolytic. If it is between 2 to 5%, it is slightly hemolytic. If it is more than 5%, the particular material is hemolytic. From this data you can understand, so almost this first sample 1 and 2, these are less than or equal to 1. That means the sample 1 and 2 are non-hemolytic, and sample 3 we can see it is slightly above the 2. That means it is slightly hemolytic. Let us see how to study the hemocompatibility of nanofibrous scaffold using hemolysis assay. In this study, we made nanofibrous scaffold using silk protein, silk sericin and silk fibroin, and this nanofibrous scaffold are loaded with SSD, that is a silver sulfur diazine drug. And this nanofibrous scaffold is mainly useful for wound dressing application. To study the hemocompatibility of this nanofibrous scaffold, we incubated this nanofibrous scaffold in presence of RBC, and after incubation we measured the absorbance at 541 nanometer. You can see the percentage of hemolysis. Our material is highly biocompatible. You can see the hemolysis percentage is less than 2%. That means the particular material is compatible. Whereas in case of water, you can see that complete hemolysis is there. This is your positive control where you can observe the complete hemolysis. Whereas in case of the test material where we use different nanofibers, you can see that there is no hemolysis. That means this material is hemocompatible for biomedical application. The hemolysis assay protocols may vary with difference in blood sources. For example, if you are collecting the blood from human or rat or goat, or it may be also vary with respect to the erythrocyte concentration, and depends on whether you are using whole blood or you are using isolated RBC, and also it depends on the wavelength which was used for hemoglobin measurement and the detergent which you use for your positive control. For example, in our study we used water as a positive control for hemolysis. In some of the studies we can use Triton X or Twin 20 detergent as a positive control for hemolysis. I hope you understood what is hemolysis assay. Let us go to lab and learn this technique more in detail. Today we are going to learn how to perform a hemolysis assay for the synthesized BSA nanoparticles. For performing the experiment, we require BSA nanoparticle stock solution, trisodium citrate treated blood. Here you can see it is not coagulated, 0.9% saline, deionized water, centrifuge tubes, microcentrifuge tubes for carrying out the experiment, centrifuge, micro tips, micropipette and vortex mixture. To proceed with the experiment, we require diluted blood. For that, we already have trisodium citrate treated blood, saline to dilute the blood, and an empty centrifuge tube to prepare the liquid solution, vortex mixture, and 1 ml micropipette, microtips. Add 100 microliter of blood in 9.9 ml of saline solution. Once we add 9.9 ml of saline, we have to vortex it to ensure that complete mixing of blood with saline. We have prepared the dilution of blood, and now we will prepare the test material. Here we will prepare 1/10 concentration of BSA nanoparticle stock solution. For that, we will add 1 ml of stock solution with 9 ml of saline. Vortex it to mix well. We have prepared the solutions, and now we are going to perform the experiment. For that, we require 9 microcentrifuge tubes, 3 sets of triplicates for each sample, where one is for negative control, second is for the test sample, and third is for the positive control. In negative control tube, we are going to add saline with blood. In test sample tube, we are going to add our test material, whereas in positive control we are going to add the deionized water. Now we are going to add 100 microliter of blood in all 9 tubes. After adding the diluted blood, we are going to add 0.9 ml of saline in the first set of 3 tubes. Then we are going to add 0.9 ml of prepared test material in the second set of 3 tubes. Finally, we will add 0.9 ml of deionized water in the last 3 tubes. Then we have to incubate all the tubes for 2 hours at 37 degrees Celsius. After incubation, we have to centrifuge all the tubes at 2000 rpm for 10 minutes. First, we are going to place the negative control tubes. Here we can observe the pellet formation for negative control. Similarly, we have to do for other 2 samples as well. Now we are going to place the test sample tubes. Here we will observe whether the test sample is lysing the blood or not. If lysis is happening, it is not advisable to use in blood related applications. Here you can see the pellet formation at the bottom of the tube, indicating that it is not lysing the blood. Finally, we are going to place the positive control tubes for centrifugation. Here you can see the lysis of blood because of deionized water. We can observe that there is no pellet formation for positive control, which confirms the lysis of blood. In the negative control and sample tubes, you can see the pellet formation, whereas in the positive control, there is no pellet formation. Now we will take 100 microliter of supernatant from all the tubes and add it to a 96 well plate for measuring the absorbance. Here we should carefully remove 100 microliter supernatant from all the tubes and add that in the 96 well plate. We have to use separate tips for each tubes. Now we have added 100 microliter in 3 columns. First column is for negative control, second column is for test sample, and third column is for positive control, all in triplicates. Finally, we will measure the absorbance at 541 nanometer and calculate the value to find out the hemolysis percentage. The hemolysis percentage, which is less than 2%, indicates that the BSA nanoparticles are highly hemocompatible and suitable for use in drug delivery or blood related applications. As a summary, in today's lecture we learned about what is biomaterial and biocompatibility, and also we learned about various hemocompatibility tests, and under the hemocompatibility test we learned hemolysis assay more in detail, both theoretically as well as practically through demonstration. Thank you for your kind attention. I will see you in another interesting lecture.